Okay, so without further ado, I will be presenting Dr. Samuel Corless. He is a postdoc in Sylvia Earhart's lab at now Karlsruhe Institute of Technology. Previously, they were at the University or Heidelberg University. Sam got his PhD with Nick Gilbert's lab, and since then he's done a lot of really great work on chromatin and centromeres, as well as DNA supercoiling and everything. So he has a lot of really great experiences, and he's won several different fellowships and awards, including like fellowships for attending conferences, as well as for his PhD and master's degrees. And I'm really excited to hear his work today from Dr. Corliss's postdoc work. So without further ado, again, I guess I'm repeating myself, I will give the floor to Sam. Thank you so much.
Okay, thank you for that introduction and for giving me the opportunity to present my work here today. I'm really excited to share with you all a new approach we've developed in order to understand the protein composition of a locus of interest, with a particular focus on the centromeric chromatin. So the centromeres, which are marked here in red, form the primary constriction on the metaphase chromosome, and are the site of kinetochore formation, and it's where the microtubules associate in order to allow the chromosomes to segregate correctly during anaphase. And so problems in centromere chromatin structure can lead to genomic instability, aneuploidy, and various kinds of metaphase chromosome defect. And so in order to understand normal anaphase, it's really important to have a good understanding of centromeric chromatin. Now, centromeres are quite unusual in that a domain of the genome which is important is not, such as, for example, a gene promoter, is normally unique sequence DNA, and it's normally evolutionarily conserved. But centromeres are built upon highly repetitive DNA that show very little sequence conservation between species. So for example, in human, we have the alpha satellite arrays, which are composed of 171 base pair monomer arranged in tandem into a higher order repeat, which is then arranged in tandem and can cover up to several megabases of DNA on a single position on each chromosome. And because centromeres are highly repetitive, and because they cover such large domains, they've been quite difficult to work with using genomics-based approaches. So for example, the human genome initially didn't contain any centromeric information, and this is also the case in HG19, which is still commonly used. And it was only with HG38 that models of centromere sequence diversity were included in the genome build. And it's only very recently that the first complete human genome, which includes end-to-end sequencing of centromeres in the correct linear order, has been put onto bioRxiv, and it's not yet published. And so because the human genome builds haven't typically contained centromeric information, our understanding of centromeric chromatin has been stuck in the pre-genomics era for the most part. This doesn't mean that we don't understand lots of important things about centromeric chromatin, but these have mostly been identified by immunofluorescence and microscopy-based approaches. But what we know is that upon the alpha satellite DNA, there's a diversity of chromatin environments within the centromere. So firstly, there's the region of sen chromatin, which is enriched for CEMP, which is the centromere defining histone H3 variant that epigenetically defines the exact position of the centromere. And it's also the protein which defines where the kinetochore attaches. And surrounding this are the largest domains of heterochromatin on most chromosomes, the pericentromeric heterochromatin, and this contains HP1 and modifications such as H3K9 trimethyl. And there are other domains which are less well characterized but have their own protein abundances. Now, because the centromere sequences were not included in earlier genome builds, when chip sequencing was developed in order to identify where a protein of interest associates in the genome, it wasn't possible to identify if it was at the centromere. So most studies which have performed chip sequencing, particularly earlier ones, really had no possibility of identifying whether their protein of interest was centromeric. More recently, since the centromeres were included in the genome build, it's mostly been centromeric labs studying known centromere proteins by targeted analysis that have used chip sequencing. And to date, nobody's really used chip sequencing to discover novel centromere proteins, and that's what we set out to do. So to do this, we developed an approach which we call REDPOINT for reanalysis of every dataset of positions of interest. And what we did was we downloaded all of the published chip sequencing data in human, which at the time we performed this analysis was just over 40,000 samples. And we aligned to the HG38 build of the human genome, which contains centromeric information. And it's important to reiterate that most of these samples, when they were originally analyzed, would have been aligned to earlier genome builds, which contained no centromeric information. And so we're identifying completely new information by recycling other people's data. And what we do is determine the percentage of reads at the centromere compared to the rest of the genome. And firstly, we can rank the data. So we have the most enriched proteins, the samples that have the highest level of centromeric enrichment. But then we also, to get greater detail, annotated just over half of these samples to include the protein target, whether it was the chip itself or a corresponding input or control, what cell type or tissue this was performed in, and whether there was a drug treatment or other perturbation. And we did this in a way that was consistent across all of these samples. And so then we could calculate the enrichment of each sample compared to its corresponding input from the same study. And we identified 307 samples that were enriched at the centromere more than twofold. In addition to looking at the centromere, we also investigated other loci by repeating this process, including other repeats and some unique sequence regions. And I'll discuss this a bit more later on. And so from these 307 samples, we identified that there were replicates of certain proteins, and some proteins, when we went back to the original data set, there were inconsistencies in that data set that we could then exclude those proteins. And we were left with 34 candidates for bona fide centromere enriched proteins. And this included in black, well-established components of the centromeric chromatin, and in blue, completely new candidates, which we can investigate as potential centromere proteins. And if we look at what we have here, firstly, we have key components of the chromatin. As a reminder, this is what interacts with the kinetochore, including CEMP-A, CEMP-B, and CEMP-C, which are the three key proteins for this region. We also have key components of pericentromeric heterochromatin, as well as a new protein, TRIM28, which is also known as CAP1. And this has been described to be a pericentromeric protein in mouse, but not previously explicitly shown in human. And for our own interest in the lab, other people in the lab are working actively on the role of non-coding RNA derived from centromeres. And this has been shown in many organisms to have an important role in centromere structure and function. But there are no known regulators of centromeric transcription, either positive or negative. And so these candidates are really quite interesting to follow up in that regard. And so in order to confirm that our approach works well, we need to check some of these novel proteins. So we just focused on two at the start. And what we see is firstly TRIM28 or CAP1, which as I previously mentioned, binds to the DAPI-dense chromocenters, which are the pericentromeres of mouse, and it's been shown in several studies. Also binds to centromeric chromatin in human. So we overexpressed CAP1 EGFB, and it forms these big foci. And we did this in a cell line which has a SNP labeled CEMP-A. What we can see is that the CAP1 foci is in the same place, but is considerably bigger, consistent with being part of the pericentromeric heterochromatin. The protein that was that had not previously been described as a centromere protein, but was the most enriched of these candidates, was JDP2. And JDP2 is not a particularly well-studied protein, but it has a number of very interesting characteristics. Firstly, it regulates histone acetylation, and it may be that histone acetylation is quite critical in the organization of mitosis and metaphase chromosome structure at the centromere. It's also an oncogenic transcription factor, and it can replace OCT4 in the Yamanaka factors in order to induce pluripotency. And if we look at the distribution on metaphase chromosomes, we can see that in blue here, the JDP2 GFP is clearly localized to the primary constriction on these chromosomes, immediately adjacent to the CEMP-A region in red. Now, it's quite interesting to note that the JDP2 appears to only be enriched on one side of the SEMPE region, whereas the pericentromeric heterochromatin extends into both sides. And this is a distribution that hasn't been explicitly shown for any proteins in human centromeric DNA. But in Drosophila, it has been described as the cohesion domain. And there are a small number of proteins which have been shown to have exactly this distribution. So it's interesting to consider whether this is another conserved domain of centromeric structure. And this is something we're looking into. So what we can see is that using our REDPOINT approach, which is a big data approach, we can identify new proteins associated with the centromeric locus. And identifying new proteins at a locus of interest has been a key, something that's an experimental approach is called locus-specific mass spectrometry has been trying to achieve for quite some time. And in locus-specific mass spectrometry, you enrich for a locus of interest using a hybridization capture approach, such as with biotinylated oligonucleotides. And then you take the protein associated with your locus of interest and you run it through a mass spec. And this is in theory a really great approach because you can identify in an unbiased manner the proteins associated with your locus of interest. But it has a number of inherent caveats. So each sample requires billions of cells. And the reason for this is that the mass spectrometer requires a minimum amount of protein in order to accurately determine the composition. But there is no technique to amplify peptide information before adding it to the mass spectrometer. And so in order to take a relatively small locus and have enough protein, you need billions of cells. This technique also identifies protein abundance rather than specificity, and it's expensive and technically challenging, and probably will never be performed outside of a highly specialist lab. But there are two highly specialist labs which have performed this technique for alpha satellite DNA in order to identify centromeric proteins. So we can see how our big data approach compares to this more experimental method. But if we consider the advantages and limitations of our REDPOINT approach, a significant limitation is that we can only use available data sets, but currently there are more than 1,200 proteins and modifications in our analysis. And this increases every day, so this limitation becomes less limiting with the publication of more data. A big advantage is because we have PCR in order to amplify the DNA, our approach is highly sensitive, and so we can use patient data, we can compare many, many cell types and conditions, and we could even consider going down to single cell analysis. It also identifies specificity rather than abundance, and it's straightforward and cheap and can basically be applied by any lab. So if we look how our REDPOINT approach compares to these published locus-specific mass spectrometry approaches for centromeric proteins, we see that there's a relatively small overlap, but that the proteins which are found in all three approaches are key centromere proteins, including CEMP-B and CEMP-C. And we also see the overlap with high-cap pericentromeric proteins and with C-burst, some of our novel candidates. And this is quite interesting because it's kind of a partial validation of our REDPOINT approach. But if we look at the proteins that we find by REDPOINT, but are not found by locus-specific mass spec, we identify interesting centromere proteins, including some of our novel candidates. But most importantly, we see that CEMP-A is in our approach, but not in the locus-specific mass spec. And CEMP-A is, again, the centromere defining histone H3 variant. And the fact that we pick it up and they don't in these mass spec approaches is probably because this protein has relatively low abundance in the centromere, but it binds with extremely high specificity and it has a very important role. So we can pick up this kind of protein, which will be missed by mass spec approaches. And I think this is another sort of advantage of our approach. If we look at the proteins that are known, have a known centromere function, which are found in the mass spec approaches, but not by us, we can see that almost all of these are in italic, and these are proteins for which there's no chip data currently available in our REDPOINT analysis. But many of these factors will probably be chipped in the near future. Some of them may already have been chipped since we did this analysis. And it may be that people in the audience today are already chipping some of these. And so, although we can't capture all of the protein diversity of the centromere with the approach as it stands, it's going to improve every day with every data set that's published. So we focused here on the centromeric regions, but I also mentioned that we looked at other loci And for a comparison with locus-specific mass spec, this is quite important because locus-specific mass spec works quite well for enormous loci such as centromeres, but it works much less well for smaller repeat regions and particularly for unique sequence regions because as the locus gets smaller, the background gets higher. And so we wanted to know, how does our REDPOINT approach, how does it work over different orders of magnitude, and is it possible to identify the proteins associated at different scales? And so we've already seen at centromeres, which make up about 60 megabases of the genome, that we have key proteins enriched, and we also identify interesting novel ones. If we look at ribosomal DNA, which is about 1/50th of the size, we again find well-established proteins involved in ribosomal DNA organization. In addition, we find new proteins which may be interesting to investigate. If we look at TAD boundaries, which are now at 14 kb, this is just a single TAD boundary on chromosome 8, we can find the key proteins involved, CTCF and components of the cohesin complex. And if we go down to 500 base pairs, we find that we have RNA polymerase subunits. And so it's possible to use this approach at any scale that you're interested in. And the way that we did it, we downloaded a subset of the data in order to look at centromeres. But if you wanted to look at these smaller regions, you couldn't download the entire data set, and then you would get even more accurate and more interesting results. In addition to identifying the static composition of a locus of interest, we can also go deeper into the data to identify data sets which show dynamic changes in protein composition at centromeres. I'm just showing one example here, but we have many such examples. And so in this case, we can see that between asynchronous and mitotic cells, we have an increase in the acetylation of the centromeric regions in mitosis, whereas there's no change in K4 trimethyl, K27 trimethyl, or K36 trimethyl. So this indicates that potentially acetylation is having an interesting role in mitosis. And as I said, we have other data sets showing all sorts of things that are changing at centromeric regions upon various perturbations. We can also perform meta-analyses of data sets that have been of particular proteins or modifications that have been chipped for many times. And this just shows, by looking at all of these different data sets from different histone modifications, something that's very well established, which is that the centromere is extremely highly enriched for H3K9 trimethyl, which is associated with paracentromeric heterochromatin, much less enriched for these other modifications. And finally, and we hope most excitingly, we can also look for drugs which target centromeric chromatin composition. And I'm not going to go into any depth on this today because we're still actively working on this, but what we have is 96 drugs and their target proteins which have been investigated in our REDPOINT analysis, either by looking at changes in the histone modifications or changes in the protein target of the drugs. And what we aim to do is to identify drugs which function through changes in centromeric chromatin structure and function. Because at the moment, there are no such drugs which directly target centromere proteins like CEMP-A, CEMP-B, or CEMP-C. They instead target proteins that are involved in signaling. But it would be a very interesting way to disrupt mitosis to directly affect centromere biology. And we have a very, very interesting candidate which appears to be doing that. And so in summary, REDPOINT is a novel approach, a big data approach for determining the locus, the specific protein composition of a locus. It can identify both the typical composition and dynamic changes. It works as loci as small as 500 base pairs and as large as 60 megabases, which covers, I think, everything that a chromatin biologist is likely to be interested in. And in current work, we're characterizing a centromere targeting drug, which we identified using this approach. So with that, I'd just like to thank the people involved, the members of Sylvia's lab, especially Alex, who was really important in some of the GFP fusion protein work. Simon Anders and Noor Pratap Singh were really crucial in helping to develop the REDPOINT analysis from something where we looked at a few hundred data sets to ones where we looked at tens of thousands. And Anna Benito's lab have contributed important work, but they also hosted me for 6 months when our lab was closed due to a flood. So I'm very grateful to them. And this is our new home in Karlsruhe. So if anybody would like to come and visit, we're here in the grounds of Karlsruhe Castle. And with that, I'd just like to take any questions. Thanks very much.
Thank you so much, Sam. That was fantastic. We already have questions, so I'm actually going to allow the audience to ask their questions, but maybe I can bug you during the coffee chat.
Okay.
So the first, oh, and I think I forgot to mention in the introduction with all the announcements, that if you would like to raise your hand to ask your question vocally, please feel free to do that. But we have a couple questions already in the Q&A chat box, so I'm going to ask those real quick. So the first question comes from Ben Weekly, and he says, really fantastic talk. I agree. Using REDPOINT, do you find these novel factors at all centromeres, or are some specific to specific chromosomes?
Yeah, that's a really interesting question. So in the REDPOINT analysis itself, we don't keep this information, but we have gone back on some of these key candidates that we're interested in to check if we see chromosome specificity. And so far, we haven't identified a chromosome specific factor. But in theory, this is possible. And for example, if this is something that somebody was interested in, it would be possible to rewrite the approach in a very straightforward manner in order to look at a particular chromosome compared to the other chromosomes. So yeah, it would be, we haven't found it, but it probably exists.
Cool. It looks like we're actually already at 9:25, but there are other questions. So please, Sam, if you have time to take a look at them, there's a question from Jason Fan in the Q&A, and a question from Alan Underhill in the chat box. So if you want to go ahead and take a look at those. But thank you for a fantastic talk. And again, if you have more questions, please feel free to join us in the coffee chat. And I'm going to give the floor to Ben.